mutation is a change in the sequence of nitrogen bases in the DNA. An error in the nitrogen base sequence can be gene level or chromosomal level. Gene mutation or point mutation is a change in one or few nucleotides in a single gene, while chromosomal mutation is a change in the structure and expression of the genes in the chromosome. A point mutation can be due to base pair substitution and base pair addition or deletion of nucleotides. In base pair substitution, a base can be replaced by another base. Base repair substitution can result in missense, nonsense, and neutral or silent mutation.
Missense mutation occurs when the coded amino acid is functionally different from the original. This stage usually has harmful effect because of the change in the structure and the function of the protein. Nonsense mutation occurs when the coded amino acid cuts short the translation of mRNA. Like missense, the nonsense mutation can be dangerous to the function of the protein. Neutral mutation occurs when the coded amino acid is functionally similar to the original amino acid. Silent mutation occurs when the change in the base sequence did not affect the structure and function of the protein.
In base pair addition, a new nucleotide is introduced in the sequence of DNA, while in base pair deletion, a nucleotide is removed from the sequence of DNA. This change creates different proteins and can be harmful to the organism. A change in the reading frame is called frameshift mutation. Alteration in the location of the gene in the chromosome results in chromosomal mutation or chromosomal rearrangements. Examples of chromosomal mutation are translocation, inversion, deletion, and duplication. Translocation is the movement of the segment of the chromosome to a non-homologous chromosome. As you can see in the image above, the segment DEF non-homologous chromosome is carrying or bearing the WXYZ segments. Inversion is the transfer of the chromosome segment within the chromosome. Deletion occurs when a segment of chromosome without a centromere is lost during cell division. Lastly, duplication. Duplication occurs when a chromosome segment is copied and joins a homologous chromosome.
As shown in the image, segment DEF was removed during deletion and segment FGHI was duplicated in the chromosome. Mutation can also be described in terms of the type of cells being affected. If the change in DNA sequence occurs in the body cells or autosomal, like the skin, it is called somatic mutation. If the affected DNA is in the sex cells or gametes, it is called germline mutation. During the cell's life cycle, errors in the nitrogen base sequence can happen spontaneously. The errors that occur during DNA replication, repair, or recombination are called a spontaneous mutation.
Spontaneous mutation is an error in the DNA sequence that occurs when a cell's error-correcting system of the cell fails to repair this error. Unrepaired errors like this can be fewer than 1 in a billion nucleotides per generation. Slip mispairing is the second source of error in spontaneous mutation. This event happens when a misalignment occurs during the homologous pairing of chromosomes. Under normal conditions, cells can revert the misalignment and return to their correct pair.
But if the error-correcting system of the cell detects the misalignment and slices the loop, it will delete several hundreds of nucleotides in the chromosome. The deletion of the nucleotide changes the reading frame, resulting in a frameshift mutation. The cell repairs damage in the DNA by nitrogen base removal, mismatch repair, and nucleotide removal. When the language in the DNA is changed, the language for making proteins is changed too. The alteration of the sequence of amino acids within proteins can be fatal or can result in the formation of new traits. The new trait increases diversity among organisms.